Hello and welcome back to Kitchen Science. So last lesson we looked at how we would produce a Petri dish that had a nice growth of bacteria on it. And today we're going to look at what we could do then once we have that bacteria grown. So once you've incubated your plate for a few days, maybe even up to a week, what you should have, if you've done everything correctly, is a nice even colouring of the agar jelly in your Petri dish that is one single colour. So different types of bacteria, different species will have different colours. So if you have one completely uniform colour, you know that you have not contaminated your sample and you only have one species of bacteria. If you did contaminate it, it might look something like this. So you might have some like bright yellow spots, you might have some orangey ones with the kind of straw coloured ones that you wanted. If you're really bad, you might even have some mold growing on it. But really what you want is one nice even growth of one species of bacteria. And once you have that, what you can do is you can use that to find a disinfectant or an antibiotic that is effective against that bacteria. And so what you could do, you'd use one plate and you'd divide it up into quarters. So you would use a marker to draw these lines on the bottom of a Petri dish in real life rather than just drawing them on paper like this. So on the underside of the Petri dish you draw in a marker lines to divide up different sections. And then along that rim of the Petri dish on the side you would write sort of what different disinfectant or different antibiotic you were going to test in each section. So maybe in this section I would test I can't think of anything now. TCP, stuff you quite often use for cleaning out wounds. And maybe in this one we'd use a 70% alcohol disinfectant. In this one we'd use bleach. And then in this last one we would use a control, and so we'd use distilled water for this. The reason why you need the distilled water is because what we're going to do is we're going to take little discs like this of filter paper. So normally it would be white filter paper. I'm just using pink so that it stands out a little bit better on my page. And you take it and you'd have it in tweezers and you dip it in your disinfectant and then you'd place it onto the agar jelly. But you want to make sure that it's the disinfectant that you're dipping it in that is killing the bacteria and not just the piece of paper itself. So if you take another piece of paper, no, I'm struggling to pick them up, there we go, another piece of paper and dip it into distilled water and you put it in this section, then you will know that if the bacteria aren't killed in this section, then if any die in these sections, it was based on what the paper was dipped in rather than the paper itself. So with each different disinfectant you'd want to test, you'd dip filter paper into it and then you'd put your little disc in that section. And then you put it back into the incubator and you'd give it time. And eventually what you would see, just write my labels on again, distilled water, 20% alcohol, and bleach. What you would see would be something like this, where you have your little paper discs with the disinfectant on, and then you have a clear area around the disc where there's no bacteria growing. This we call the zone of inhibition, because the disinfectant has inhibited or stopped the bacteria growing in that area. And the bigger the area around the circle, around the disc, the better the disinfectant is at stopping that bacteria from growing. So if we look down here at distilled water, there's no clear area around it. It is not effective at all. So we definitely know that any of this bacteria that is being killed in these sections. So it's definitely down to the disinfectant, not down to the piece of paper. We've got a bit of a clear area around the TCP, a bit more around the 70% alcohol, and a bit more around the bleach. So initially, just by having a look at these, we could say that probably the bleach is the most effective disinfectant against this type of bacteria. Obviously, every different species of bacteria, you might find you get slightly different results for, but for this bacteria, the bleach is the most effective. But we can go further than just looking at it and drawing general conclusions. We can actually say how much better it is. And we do that by calculating the area of the zone of inhibition. Now, hopefully in maths, you've talked about working out areas, and usually the zone of inhibition tends to be a circle. It might not always be a perfect circle in real life, and so you'll have to kind of guess where roughly the diameter, the radius would be. But in exam questions, you are most likely to get given perfect circles. So hopefully we are happy with the idea that the area of a circle is equal to pi r for radius squared. So the first thing that you need to do is work out the area of your circles. So work out the radius of your circles. So I'll start with the TCP one, and it's usually easiest to measure the whole diameter and then divide by 2. So I've got a diameter on this one of 20 millimeters, so a radius of 10 millimeters. So the radius equals 10 millimeters, and the area be equal to pi times 10 squared. And I'm going to use my calculator. So pi times 10 to the power of 2 equals, probably should have been able to work this one out in my head, but oh well, 314 millimeters. Sorry, my recording got all mixed up, and so I've gone and done all the answers, and now I'm going to have to go over it again and explain to you how I got these answers. So for the alcohol, measure it, we got a diameter of 30 millimeters, it's a radius of 15 millimeters. So for my area, we do pi times 15 squared, which gave me an answer of 707 millimeters squared. So all of these I am working out to the nearest millimeter. So for the bleach, diameter of 44 millimeters, so an area of 22. So a radius of 22, my area is pi times 22 squared, which gives me an answer of 1,521 millimeters squared. The advantages of working out the areas mainly is if you had zones of inhibition that weren't obviously different sizes, measuring and calculating the area will just be a little bit more precise and help you to say which one is definitely the most effective disinfectant. This is a required practical, so it's something that we will do when we are back at school. So we will go over it again then. However, a little bit of information on the things that you'd have to keep the same, so your control variables, to make sure that this experiment is fair. You'd have to have the same volume of disinfectant on each disc. You'd have to have the same radius of the disc itself. You'd have to have the same concentration of disinfectant in each solution. You would need to have if you were using 2 separate Petri dishes, if you were wanting to test more disinfectants, you'd have to make sure that the Petri dishes were kept in the same conditions, that they were incubated at the same temperature for the same amount of time, that they have the same amount of nutrients in the agar, the same amount of oxygen available to them. All of these things will affect bacterial growth, and you want the only thing different to be the type of disinfectant that is on that disc. Now I'd like you to have a go at doing some calculating zones of inhibition yourself. If you have any questions, get in touch with me and ask, and I'll do the best I can to help you. Bye.